Hello, thanks for coming. My name is Griffin. I'm a fourth year graduate student in the lab with Professor Joe Wedekind at the University of Rochester, and today I'm very excited to tell you about the discovery of a small RNA that cooperatively senses two stacked metabolites in a single binding pocket for gene control. I'll be focusing today on the preQ1 riboswitch family, which recognizes preQ1. preQ1 is the last free intermediate in the quercetin biosynthesis pathway. This begins with free GTP, which is converted enzymatically into preQ1. preQ1 is then inserted into the wobble position of GUN anticodons on tRNA, where it is later modified into quercetin. And quercetin really aids in codon-anticodon pairing, but its importance in maintaining homeostasis is really highlighted by the three classes of riboswitch that regulate various aspects of this pathway. Now, a representative of each of these has been characterized extensively by our lab and others, which has revealed that these three classes are very different in terms of their size and the folds that they adopt. However, they are all known to bind one copy of preQ1 to regulate quercetin production. Of these three classes, the best studied is certainly the class 1. Its small size has facilitated many in vitro and in silico studies and is also the most prevalent among pathogens. It turns out that the class 1 riboswitch can be divided into three subtypes, and all of these are predicted to form an H-type pseudoknot based on covariation data published by the Breaker Lab. Shortly after this prediction, two crystal structures of class 1 riboswitches from Thermoanobacter tencongensis and Bacillus subtilis confirmed the H-type pseudoknot fold, and with high-resolution structures in hand, these two switches became models in the field. However, based on sequence analysis, it is apparent that both of these are actually type 2 sequences. And so despite this family being known for about 15 years now, there is still very little information about the type 1 and type 3 subtypes. Even though each of these is predicted to form an H-type pseudoknot, the covariation models differ at key positions near the preQ1 binding pocket, which suggests that there might be subtle differences in preQ1 binding between these three subtypes, which could lead to better insights to develop small molecules to drug these riboswitches. And so I decided to further explore type 1 sequences because they are the most prevalent among bacteria, with just under 1,600 known representatives. Now, to investigate whether these differences in the covariation model actually lead to differences in preQ1 recognition, I solved a co-crystal structure of a type 1 riboswitch bound to preQ1 at 2.6 angstroms resolution. The riboswitch I used was from Carnobacterium antarcticus, which I will refer to as the CAN riboswitch today. At first glance, the fold of the CAN riboswitch appears quite similar to a type 2 structure from Thermoanobacter tencongensis, which is here on the right. And this is consistent with that prediction from the Breaker Lab, as the CAN riboswitch indeed adopts an H-type pseudoknot fold. However, my structure unexpectedly reveals two preQ1 metabolites stacked on top of one another in a single binding pocket. These two metabolites are coordinated by an elaborate network of hydrogen bonds, including an unexpected preQ1 to preQ1 hydrogen bond. For the rest of this talk, I'm going to split this single binding site into two binding subdomains just for the sake of clarity. The alpha site will refer to this more brightly colored preQ1 here on the bottom of this diagram, while the beta site will refer to this more darkly colored preQ1 here on the top. From my structure, it is clear that specificity at each of these sites is conferred by a cytidine. For the alpha site, this is done by C17, which is engaged in 3 hydrogen bonds with the Watson-Crick face of the alpha preQ1, while at the beta site, this is done by C31, which is engaged in 2 hydrogen bonds with the sugar-like edge of the beta preQ1. Now, it's important to point out that riboswitches that sense more than one metabolite are really rare, and this is the first example of a dual-sensing preQ1 riboswitch. Importantly, when we go back to that covariation model that I showed you earlier, each position in the alpha and beta binding site is 97% conserved. And so this strongly suggests that this extraordinary mode of preQ1 recognition, which is unprecedented in RNA biology, is a hallmark feature of the type 1 riboswitch. And so I really wanted to confirm this binding stoichiometry using another experiment. And to do this, I used isothermal titration calorimetry, or ITC. This is a label-free method to determine how tightly a ligand binds a receptor. And an ITC experiment directly provides 3 pieces of information: the change in enthalpy, a binding constant K, and an N value, which we are interested in because the N value reports on binding stoichiometry. The ITC thermogram I'm showing here reveals that preQ1 binding to the CAN riboswitch is highly exothermic and is best characterized by a KD of around 30 nanomolar. What's really important though is this N value, which again is the stoichiometry, which is 1.8. This number strongly supports the 2-to-1 interaction that I observe in my structure. Now, it's also important to point out that this high N value combined with the single phase shape of this fit suggests that this riboswitch uses positive cooperativity to recognize these two metabolites, which is yet another rare riboswitch trait. I explored this further by repeating these RTC experiments at a more physiologically relevant temperature of 37 degrees Celsius. When I did so, the shape of the binding isotherm actually changed, and I was able to fit two KD values, with the second being more favorable than the first. I was able to use these two KD values to calculate a cooperativity value, which was a positive 7.7, further demonstrating this riboswitch uses positive cooperativity. We independently validated this by collaborating with the Schroeder lab to use a REL-E-based cleavage assay, which you can hear more about from Carolyn Focht, who is also giving a presentation at this conference. Her program number is C1404. And what Carolyn found is that this riboswitch inhibits ribosome assembly with added pre-Q1. It has a K1/2 of around 15 micromolar, but importantly a Hill coefficient of 1.4, confirming that this riboswitch is cooperative. So at this point, my structure and functional data reveal that the CAN riboswitch cooperatively binds two pre-Q1 metabolites in a single binding domain. Moreover, this mode of recognition is a hallmark feature of the most prevalent and widely distributed pre-Q1 sensing riboswitch. And so with this in mind, I decided to change gears and ask, how does this unusual mode of metabolite recognition lead to gene regulation? Does binding two metabolites make this riboswitch a more effective switch? And are both binding events actually necessary for regulation? To answer these questions, I use an in-cell reporter assay that our lab has used previously, where we design a plasmid where we place the riboswitch in the 5' leader sequence of a GFP protein. We transform this plasmid into QF knockout E. coli cells, which are pre-Q1 deficient, and conduct all of our experiments in a minimal media that has no pre-Q1 in it. Over the course of the experiment, I will add pre-Q1 to the media, which is shown here on the x-axis, and monitor GFP fluorescence, which is shown on my y-axis. On this graph, the light blue line corresponds to the CAN riboswitch. As you can see, with added pre-Q1, it clearly represses the expression of GFP. This is best described by a biphasic curve with EC50 values of 96 nanomolar and around 7 micromolar. The other two curves on my graph are my controls. My positive control is this green line here. This construct contains a GFP gene, a Schroeder sequence, but no riboswitch, so it should always express GFP. And likewise, my negative control, which is this red line here, contains a GFP gene, an inactive Schroeder sequence, and no riboswitch, so it should never express GFP. As a proof of principle, in levels of low pre-Q1, the CAN riboswitch permits levels of GFP expression that are comparable to my positive control, but as pre-Q1 increases, it will knock this down to be at levels comparable to my negative control. It is worth acknowledging that the biphasic shape of the wild-type curve is a little bit unexpected, but we can still analyze how well the riboswitch works by calculating full GFP repression. As we predict, my control showed very little GFP repression with added pre-Q1, while the CAN riboswitch demonstrates a 15-fold repression ability. And this is actually remarkably similar to the repression that we saw in a class 2 pre-Q1 riboswitch from Lactobacillus rhamnosus, which we have studied extensively and binds only one copy of pre-Q1. So it appears then that even though the CAN riboswitch binds two pre-Q1 metabolites, it is not a more potent off-switch than one that binds only one copy. Now, to ask whether both these metabolites are actually necessary for gene regulation then, I decided to mutate the main specificity bases in each of the binding subdomains. Just as a reminder, this would be C17, which will impair binding at the alpha site, and C31, which will impair binding at the beta site, and it mutated each of these cytidines into uracils. And both these mutants fared really poorly in this experiment. C17U was the better performer of the two. It maintained a 6-fold repression ability, while C31U was really no better than my controls at only a 2-fold. And likewise, C17U showed a more mild increase in its EC50 of around a 60-fold increase, while C31U suffered an over 200-fold increase. And so what these two results together reveal is that both pre-Q1 binding sites must be fully functional for effective gene regulation. So I hope I've convinced you I've identified a riboswitch that binds two pre-Q1 metabolites in an unprecedented way, in which the two metabolites cooperatively stack on top of one another in a single binding pocket, and that both these metabolites are required for gene regulation. Our current working model is shown here on the right, where in the absence of pre-Q1, the Schroeder sequence is free to the ribosome, so the gene is turned on. In the probably very shortly lived, partially occupied state, there might be a modest amount of gene regulatory activity, as the Schroeder sequence might be transiently sequestered, but only in this fully occupied state do we see the gene turn completely off and the Schroeder sequence fully sequestered. Now, a really exciting aspect of this project is that the mode of pre-Q1 recognition appears to be strongly conserved amongst the type 1 subtype and is utilized by many pathogens such as Neisseria gonorrhoeae to maintain homeostasis. So this new mode of pre-Q1 binding is a really exciting opportunity then to develop new bioorthogonal antibiotics to target these antibiotic-resistant bacteria. And finally, this aptamer demonstrates that an RNA can closely position two metabolites in a single binding pocket, which really expands what we think is possible in terms of substrate recognition by an RNA. Now, the high conservation of this aptamer suggests that it may have origins in the RNA world, where it may have originally positioned two substrates close together within a ribozyme for something like chemical linkage or the transfer of a chemical group. That's the end of my talk. I'd like to thank an awful lot of people for this work, specifically my PI Joe for all of his support, Dr. Jenkins for helping me solve the crystal structure, and a very talented undergraduate, Maya Blau, all members of the Wedekind lab, past and present, Dr. David Matthews and Dr. Chapin Cavender for helping me interpret some ITC data, Carolyn Focht for performing the REL-E cleavage assays, Professors Ben Miller and Clara Kilkoff for letting me use some of their instruments, and of course my funding. And with that, normally I would take questions, but since this is virtual, please feel free to email me or find me on the attendee page online. Thank you for listening.